All right, so again, good day, learners. So today we will jump to our next video tutorial, ANOVA made simple, statistic differences across multiple groups. So one-way or two-way ANOVA. So we can also run post-hoc test, which is the Tukey, and other post-hoc test. And first, we will check the assumptions whether our data is normally distributed or homo in terms of homogeneity, and also trying to look at the effect sizes and how to interpret and report the results, which is ready for publication. So without further ado, let's jump into our R. Now, again, I think I've already shown you a lot of this R Studio, but then I will try to make a new file just to be able to really like see what's going on with the code. So this is a clean code, a clean uh, R script blank. So this is where we're gonna write our code. So this is ANOVA. So this is already a code that I asked, I somehow prepared for this lecture. So let's say first is we have to load this uh, library. So I'm just gonna do the library. And again, you can always write a code in here, like I wanna say, uh, library. So, just to make sure that you are in the right direction and to be able to understand it really this ANOVA. So next is because we wanted to run a simple ANOVA. So in that case, let's try to create a simple data set. Okay. So again, this is a simple data set created from here, but then maybe next time I'll try to use actual data set that I've been working. I have a lot of data. I published more than three manuscripts this year. So maybe in one of these days I'll be able to show my real data. Anyway, so let's try to make a simple data set. So in this case, I have three forest sites. So now like why I have three sites and for my t-test, I only have two sites. So again, if you want to use t-test, you need two groups. So it could be a category, let's say for example, if for pre-exam, after exam, or pre-lecture, or post-lecture, or you could have, say, look at that the severe disturbances and the less disturbances or no disturbance or higher disturbance. So you can look at the difference of this by using t-test. But then the other thing is there is so-called ANOVA. So that's ANOVA, analysis of variance. As you can see here, ANOVA, that's analysis of variance. So here we will try to create a data set that is two groups because for ANOVA, minimum variables or groups so that you can use this as at least three groups. So if there is differences, so I think you've already observed a lot of let's say experimental design. So experiment one, two, three, and then you have the control, and then you kind of use the ANOVA. So like just to look at if there are differences among the treatments and or your control treatment. So again, this is the same pattern. So see this is how you create data, the site E, site B, site C. So we can look at it that site B is site C has the highest mean, followed by 28, and then site A 25. So let's just run this site E and B and C, and then let's say table site. We're gonna have to replace site A as A, B, C. Each is 30. So this is the number of observations that we wanted to create, and DBH site A, site B, site C. So let's say I'm gonna put here as DBH and different just to make sure that our unit is visible. So I'm gonna run that. Now I have 90 observations because we have 30 each site. In that case, we have 90 observations of two variables. Two variables means because as site A, you have DBH in each site. All right. So I think we already created data set. So let's try to run it a little bit just to look at our data. So head data, this is what you see. Again, we can see the dimension data. So we have 90 observations, two variables. We can have the string of data. So these are like basic, basic framing, visualization of your data set that you're dealing. So here, and you can see the summary of S and then data. So it's not working. Let's just change that later on. Or maybe we could do this in A that just try to uh, run everything. Okay, it's not working. I'm not gonna do that. So let's just try to look at the summary of our data. So here, because we only have one data, two variables, so site, we have 90, and the DBH, you can see DBH, which is 12, first quarter, and our mean is 28, and the maximum is 45. So that's basically what we can see, okay? So let's try to visualize this a little bit. So I'm not gonna, I think I've already explained this later, in my previous tutorial. So here, the ggplot data, again, data, make sure that what's the name of here, it should be here, and the site and dbh. So the gym plot field, sky blue, you can always change that. So let's say blue, you like blue, I like sky. And logs, I don't need to write the title, so I'm just gonna delete that. And site dbh, which is centimeters, and make sure, because sometimes um the author uses DBHS meters to make sure that the unit is visible enough. So in that case, I will try to DBH. So DBH, because so what happened is DBH not always caused by error. DBH is not possible. How is that possible? So because here we need to try to look at DBH underscore centimeter. Basically, your coding is not correct. So you have to write it that way and then rerun again, and there you go. So you have it. So like, wow, that's interesting. Like, I like this one. So you can see the from, I would say there is like a linear relationship, not linear, but just trying to look at E, B, and C. So the higher the side, the higher the dB. So we can say that side A is maybe we can, if we don't know the study side. So I would probably assume that like, yeah, maybe side A is on the lower slope, side B is a middle slope, and side C is higher slope. So we can expect that E is prone to disturbance because it's it's close to the access to disturbances, but a higher elevation, it means that maybe less people going there and something uh like that. All right, so now let's try to. Okay, I'll be there. So let me just pause this because somebody is calling me, so I'll be back. Okay. All right. So sorry about that. Now let's continue. So where are we? So I'm gonna run this again. So see, you can see that there is a pattern among the groups. So I'm just gonna say that Vax, Lot, and Nova, and I'll click save, and we'll just go directly to my file, and you can see there ANOVA box plot ANOVA. Now you are ready to copy the figure and you are good to go. And let's say can write it here and so on and so on. Then you are good to go with this. And so with that, let's try to move a little bit more forward. So first is we have to check the assumptions. So let's just try to track it here for visibility. So here, I think I've already mentioned about normality. So let's just try the very simple the test. Look at the distributions of our data set. So test the data and then dBH centimeter. So here we can say that our data is normally distributed. So you can see the pattern, normal distributions, more on the center, and I think I would expect that the Shapir test would be uh higher than 0.05, which is correct. So the p-value is 0.085, which is higher than higher. So I'm just gonna write that so we can make it visible. So our p-value is higher than 0.05. So it means that if this value is higher than our data is normally distributed. So simple as that. So let's say if this is 0.000, then that is lower than 0.05. It means our data is not normally distributed. So I don't know if you get the whole idea of this normality. So next is another assumption is the so-called homogeneity of variance. So we can try the beginning. So first is we have to load the library car. So there is no, it's working, I think. So loading, yeah, it worked. It loads perfectly. Now let's try to Linear test, DBH by side. So let me just check. Side DBH, that's injector. So I have to check this one. Make sure that your DBH is ready this way. So again, numeric here and then side. Okay, this is numeric, this is side. So data, and this is how you. So here, I'm gonna look at this one here. So the p-value, it's a numeric test is 0.29, so I think that's okay. P-value is greater than 0.05, so the samples are okay. So our data set means it's homogeneity. So now let's try the ANOVA. You see, ANOVA is very simple. So ANOVA code is very simple as this. So again, when you do the ANOVA, so a analysis of variance, and again, always numeric. And this symbol at the side data is equals to data. Close it and you are good to go. So but if you wanted to make a new file, then you can just ANOVA model and just do like this. So see it's not working. I will just say centimeter. So now our ANOVA model is here, but it doesn't make sense here. So that's why you have better to just summary of your ANOVA model. And this is the model of this is summary of our ANOVA. So you don't have to see everything there, just the significant goal. So if it's 0, that's highly significant 3 star, and 0.01 2 star, 0.01 is 1 star, but it means that they still are significant. So you can say highly significant, not significant, but here we have 3 stars. It means that our p-value is like 0.00008, something like that. So we can say that there is variation, there is significant, or we can say there is highly significant among the groups, which is basically you have seen it in our backplot. So let's try to go back to our backplot. So see, this is our backplot. But just looking at the data with our backplot, you can already see that the sites among the groups there are a lot of differences in terms of image. Yeah. So let's try to look at here. So let's say actually you can also just run like this ANOVA, and there you go. You can already look at here, and you can say, or you can say ANOVA model, and that's it. So see, that's the power of R. And now let's proceed to the Tukey post-hoc. So Tukey post-hoc is because you already know that there are differences, but then you don't know which group is significant. So in this case, so we already know the fact that there are differences among the sites, but then we don't know which sites are highly significant. So I would say maybe E and B is highly significant, or maybe E and B or E and C or B and C. So that's our assumption. But we don't know yet. So that's why we have to run our so-called post-hoc. So just the formula is just this: Tukey, HSD, and then you write the name of your model. As simple as that. So if this is model, let's say model one, and we will just model one, so model one, and you just have to write it here like this. Model one, as simple as that. Now you have your results of your model. Now I want you to look at this is the code that we ran. So Tukey, model one and site is our site A. So I want you to look at the value for P adjusted. So P adjusted is here 0.01, 0.00, 0.00 C. We can see that highly significant is C and A, which is, I would say, that is very obvious. So E and C is highly significant because look at the differences in terms of DBH. But then it's interesting to know as well that E and B is 0.01 and C and B is 0.00. So in that case, we can see that E and C is highly significant. Also, B and C is significant, but not as much as E and C and also E and B, which is visible in this box plot. So now you're ready to report. You can copy this results and to this one. I usually just uh I sometimes let's say two key. We'll try this. So two key results. So let me just do this one. If it's so see two key, no, it's not good. But I will just like uh, look at the value and copy it to Excel. Pretty easy anyway. So now let's try to look at the effect on sizes. Effects on sizes. This is just an additional. So we can say the library again, library. So if you don't know how to find library, just go to packages and just install and just type library. No, well, just type effect plots, effect size. This is the effect size. Then you click install, but I'm not gonna do that because it's already installed here. So I'm just gonna run this. And then ETA squared ANOVA model, which is I did here. So let's just try it. And it says, oh, okay, that's interesting. And so here is the interpretation. So you have the small 0.6 and 0.4, which is large. So we can say point R. So this is 0.35. So it means large. So our differences in terms of sizes is very large. So it's just like that. And then you are ready to record. Now let's proceed to two-way ANOVA. So two-way ANOVA is a different, still ANOVA, but it's another uh, analysis. So let's here we created new data set, data two. Let's just name it the data two table. Replace E, B, and C. Rep, sure replicas, H30 times 3. So we will be doing it 180, weighted mean, and this one. Okay. Now let's try to have data two. We have 180 observations, 3 variable. So species DBH. So in this case, TOEI ANOVA because we are now dealing with another variable. So the first simple ANOVA is we have only the site and DBH. All right. So E, B, C. So we don't really know like E, which is A, B, and C. But then for uh for for TOEI ANOVA is like we wanted to look at like if there are any variations of species among the sites. So that's kind of our story for running a TOEI ANOVA. So that's why here we have 3 variables. We have site A, site, which is site A. So EBC, I would speak the EBC, Sharia. We have only 2 species, Sharia and Picus. And the fish minimum 15 and highest is 39. So which is interesting for me. So now let's try to look at model 2. So let's say I'm just gonna make it model two-way, just make sure that we are understanding this in a very easy way. All right. So again, this is TOEI ANOVA. So like how was it like? It's an interaction. So it's pretty the same with ANOVA, but then we just added a species. That's the whole idea there. So data too, I'm gonna use the data. So do not be used with the data because there's no data on species. So let's say you're gonna put it data alone. So what will happen is there is so-called an error. It says object DBH not found, which is yeah, it's not true because there is DBH in here, but it's centimeters. But then um I'm gonna put it this way like this, so just to make sure that we're so see, so object is species not found, so which is correct because there is no species on our first data set. But then here is this is already data too, so make sure that your data species db is so let's make sure db is so that lit centimeter species. So just to make it that it's easy, I will just type names data two. Okay, are we following to that? I'm not sure if I'm fast, but this is it. Oh no, I don't want to save it. So names data two. Now you have site. That's correct in here. The species, that's correct in there. And DBS. Then we are ready to run our two-way model. Okay, model two-way. There you go. And then I'm gonna try to summarize the model two. Okay, model two not found, so because I changed it, so I'm gonna change it this way here, and there you go. That's interesting. So this is the result of our two-way model, and here you have the site, and basically there is no differences among the site because look at the p-value, which is 0.8, and the species as well, which is 0.1, but then the site and species, which is also 0.8, so this is higher than the site. So basically, it is there is no, I would say there is no significant among the sites and among the species, so which is a different story line. So it's like okay, so if like, there's no difference among species, there's no differences among sites. So it really doesn't matter like whether it's a ficus or a sharia or among the edges, there's no make sense. So another thing that you can do is you can do plot, but just the model to model two-way, and I'm gonna plot that plot. And here is the plot. So see, this is the first plot. So you can look at the plot for residual versus fitted. So it will take a lot of time to interpret this, but just trying to look at the results of your model, and then you just hit enter, and this is like the QQ plot, the residual. So it looks good on me. Look at the line. At the line is perfectly the data points perfectly fit to the line. And last plot is this the scale location fitted versus values. So this is just how you checked your model. All right. So yeah. So I think I can do like this as well. Here. So again, there is such thing as personal differences among the species and among the size, which is interesting because of course, we use a different set, and when you just look at it, so we basically just created a data set that is among the three species and three sites, but then like are almost the same. Yeah, the mean is 628 and the standard deviation is 5. So like, the standard deviation is 5, which is lower. I would say the data points among the individual points is not that really uh differ from other data points. All right. So I think that's very simple analysis that we can derive from there. So now let's try to look at how to report results in your paper. So let's say if this is already a paper. So I'm just gonna write this way just to make sure that we understand it. So ANOVA, so there was significant differences in mean deviation among sites, which is really visible. So again, it was P.05, and you see the large effect. So it's 0.22. And then the Tukey host post showed the side was significantly higher than side A. So we knew that. So if you try to remember our results for the post hoc here, so let me just go back in there. So here it's point 0.5, 0.01, 0.00. So see, this is the results of our interpretations. And so side A, side B, and Toei ANOVA. So although we are using a different data set, so here this significant mean effects were found for side and species 0.03. The interaction term was not significant. So the interaction term was not significant 0.4, indicating that the side was consistent across the area. So that pretty is today. So I think you learned a lot, not a lot, but at least you will be able to run ANOVA and at least interpret how to do the ANOVA, how to run ANOVA, and how to produce figures, how to look at the p-value, and also how to check the normality using Shapiro and homogeneity using the Levene test. So I think that's all for today. And again, that's all for this ANOVA made sample. So as long as we know the data, so that's why it's important that when you collect the data, you're familiar with the data. So let's say there was a significant difference among the three sizes. Like, and as a researcher, you are responsible like why there was a significant among the three groups. So let's say because I was a researcher, but yeah, because side A was really disturbed, and side C was on the top already on the mountains and higher. It was higher elevation, so it's good. I would expect that the area is less disturbed, and so the differences are higher when you compare side A and side B. So like it's important. When you write your research, it's like you know your data. You should know your data and what information you can get from it. And that's exactly the whole idea of why you are doing research. You wanted to know what made the differences, and of course, you should be able to understand what's going on in there in the field. All right, so I think that's all for today. And again, you can contact me with my email address, rodrigorafi@gmail.com, and my also work email, rafi.rodrigo@ipso.edu.ph. And with my engagements, I'm actually an editor-in-chief of the Journal of Philippine Island Universities. So if you have paper, you can please submit to your paper. So I will really appreciate if you can submit your thesis. And I am a reviewer of several papers. So with Nature Portfolio, Elsevier, and but mostly in forestry field. And yeah, so see you in the next video.